Namaste to all students. Kushbu Bidya, myself JV and Dr. Kushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about haploid plant production under PTC application session, and this session related to plant biotechnology course of MSc BT second sem program. So what is haploid plant production? So in the haploid plant production, you produces haploid plants by anther culture or by my pollen culture or by ovary or ovule culture. You can create haploid plants. So by the haploid plant production, you can embryo rescue of interspecific crosses, and you can create alloploids. And in the context of haploid plant production, you can go for anther culture or microspore culture. And in the anther culture or microspore culture, you have to culture of anther or pollen grains, and which derives a mature plant from a single microspore. On the other hand, second type is gynogenesis, which has involved ovary culture or ovule culture. So in ovary and ovule culture, you have to culture of unfertilized ovule, which are known as macrospores. What are the examples, specific examples of DH uses? So evaluate fixed progeny from an F1. It can evaluate for recessive and quantitative traits, and it requires very large dihaploid population since no prior selection, and it may be effective if you can screen some qualitative traits early. And it is for creating permanent F2 family for molecular marker development, and it is for fixing in-bird lines, basically for novel uses like creation of few dihaploid plants from a new in-bird prior to going to foundation seeds. It allows you to uncover unseen off types, and it is basically for eliminating inbreeding depressions, which is theoretical till now. And if you can select against deleterious genes in culture and screen very large populations, you may be able to eliminate or reduce inbreeding depression. For example, inbreeding depression has been reduced to manageable level in maize through about 50 plus years of breeding. This may reduce that time to a few years for a crop like onion or alfalfa. In somatic hybridization, you develop hybrid plants through the fusion of somatic protoplasts of two different species or varieties. Here I am sharing the screen of my PPT by which you can understand the somatic hybridization by a real microscopic picture. Here two protoplasts. You know very well that protoplast is a cell, plant cell, which is not having any kind of a cell wall. So without cell wall, plant cell is known as protoplast. So there are two protoplasts. Here you can see single protoplast. So similar two protoplasts come in the contact with each other and they start to fuse with each other. And first of all, they create an intermediate which is known as heterokaryon, which have mixed cytoplasm, but still two nucleus are separated from each other. And after some time, these different two nucleus also fuse with each other and they finally create hybrids. There are some process or steps which has been followed during the somatic hybridization process. So first is isolation of protoplast, second is fusion of the protoplast of desired species or varieties, and third one is identification and selection of somatic hybrid cells, and fourth is culture of hybrid cells, and fifth is regeneration of hybrid plants. So the isolation of protoplast means separation of protoplast from plant tissues, especially from cell walls. So you have to go for mechanical and enzymatic method, but enzymatic methods are having more advantages in comparison to mechanical method. If we talk about mechanical method, so you have, first of all, you have to select plant tissues, then you have to go for cell plasmolysis. After cell plasmolysis, you can do microscopic observation of the cell, and further you have to cut cell wall of the cell with sharp knife, which release your protoplast. Then you have to collect all the protoplasts, but one problem is here, like whenever you are cutting with the knife, it can rupture the protoplast or it can damage the protoplast. So at the end of the process you can get less number of viable protoplasts. In mechanical method, it is used for the vacuolated cells like onion bulb scale and radish and beetroot tissues. It create low yield of protoplast because I have told you the reason, and it is laborious and tedious process because you need skilled person, and you will get low protoplast viability in this mechanical method. Next type method is enzymatic method. So first of all, you have to go for the leaf crystallization process and removal of the epidermis, means you have to peel off your epidermis. Then you can go with the two procedures, like you have to go first of all for plasmolysis process. After the plasmolysis process, you can go for direct method or you can go for the indirect method. In the direct method, you will use all the chemicals or all the enzymes in one step only, and in indirect process you have to use enzymes step by step, one by one. So in direct method, after the plasmolysis of the cell, you have to incubate these plasmolysis cell with pectinase and cellulase enzyme, which will release the protoplasm, means they degrade the cell wall of plant cell and create protoplast. In the second type, which is indirect method, in this method, you first of all you have to incubate plasmolyzed cell with pectinase enzyme, which release the isolated cells. Further, you will incubate these isolated cells with cellulase enzyme, which further create finally protoplast, means they will finally degrade the cell wall. So that's how you can get or you can isolate the protoplast from leaf cells, and by the enzymatic method you will get more number of viable protoplast, because it is not going to damage any kind of a protoplast, and it is used for the variety of the tissues and organs including leaves, petioles, fruits, roots, coleopetils and hypopotyle stems, shoot apex and embryos, microspores. And in the mesophyll tissues, it is more suitable process, and by the enzymatic method you can get most of the number of protoplasts by mesophyll tissues. It is having high yield of protoplast, it is easy to perform, and more protoplast viability you will get. In the case of protoplast fusion, so which is a fusion of protoplasts of two different genomes. So first type is spontaneous fusion and second type is induced fusion. In the spontaneous fusion, it is also subdivided in two groups, means intra-specific and intergeneric. And in induced fusion, it is having three types: chemofusion, mechanical fusion, and electron fusion. There are some uses of protoplast fusion. It combines two complete genomes, means it is another way to create allo polyploids, and it is basically in vitro fertilization, in which you are going to fuse two different species protoplast cells, and it is partial genome transfer, means exchange single or few traits between species. It may or may not require ionizing radiation, and it is involving genetic engineering, like microinjection you can use, electroporation, and agrobacterium mediated gene transfer technique also you can use during the protoplast fusion. And it is basically used for the transfer of the organelles, like it is unique to protoplast fusion. The transfer of mitochondria or chloroplast between the species you can done by the protoplast fusion method. In spontaneous fusion, protoplasts fuse spontaneously during the isolation process, mainly due to physical contact. Intraspecific produces homokaryons. Means if you are fusing same species protoplast, so it will create homokaryons. If you are using intergeneric, So it is not having any kind of importance. In case of an induced fusion, so chemofusion is basically fusion induced by the chemical. So there are various types of fusogens which induce the fusions like PEG, NaNO3, calcium ions, polyvinyl alcohol. And in induced fusion, there are two types of a method like mechanical fusion. So physical fusion of protoplasts under microscopic by using micromanipulator and perfusion micropipette. And in case of a electrofusion, it is fusion induced by the electrical stimulation. So fusion of protoplast is induced by the application of high strength electric field of 100 kilovolt for few microseconds can create the fusion of protoplast. So there are possible results of the fusion of two genetically different protoplasts. So here you can understand there is a possible result by this diagrammatic representation. So here I am sharing the screen. There are two different protoplasts which are having two kind of different organelles. Whenever you are going to fuse it, so first of all it will create the heterokaryon. So there are two possibilities, like these are all hybrids, like when they are fusing, nucleus is going to fuse, and but if both nucleus are going to fuse, so it is hybrid. It is no matter like if they are only using chloroplast and mitochondria of any kind, any of species. Basically, hybrid concept is dependent on the fusion of nucleus or the contribution of both parents in the nucleus formation. So these two species are hybrids, results are hybrids, but these are hybrids because in this fusion, only one parental nucleus is available and the second one is not present. So that's why this kind of a final product is known as hybrid because it is having only one nucleus of one parent. It is having both parent cytoplasm, but still it is having only one nucleus. So that's why we are calling these two results hybrids and these two results hybrids. How you are going to identify desired fusions? So there are complementation selection, fluorescence, activated cell sorters, mechanical isolation you can done, and mass culture you can done. And there are some advantages of somatic hybridization like production of novel interspecific and intergeneric hybrids like tomato, hybrids of potato and tomato. Production of fertile diploids and polyploids from sexually sterile haploids, triploids and aneuploids, and transfer gene for disease resistance, abiotic stress resistance, herbicide resistance, and many other quality characters. And there are production of heterozygous lines in a single species which cannot be propagated by vegetative means. It is also useful for the study on the fate of plasma genes, and it is also used for the production of unique hybrids of nucleus and cytoplasm. But there are some problems and limitations related to somatic hybridization, like applications of protoplast technology requires efficient plant regeneration system. There is lack of an efficient selection method for fused product is sometimes a major problem. The end product after somatic hybridization is often unbalanced. Development of chimeric calyces in place of hybrids can be done during the somatic hybridization. So this is also one problem. And somatic hybridization of two diploids leads to the formation of an amphiploids, which is generally unfavorable. Next limitation is regeneration products after somatic hybridization are often variable, and it is never certain that a particular characteristics will be expressed. And genetic stability is also a problem. You cannot get genetic stability, and sexual reproduction of somatic hybrids is also a problem, and intergeneric recombination is also a problem or limitation during the somatic hybridization process. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.